Hello?
Oh, there you are.
Yeah. What's going on today?
Well, I have tried to add a few slides to your Tensegrity upload, and all the slides got dumped onto one slide.
Oh, yeah, I saw
I
it.
don't know what I was doing wrong.
Oh, well, I did get the slideshow you sent me and I put it in, so they should all be there.
Okay, what was I doing?
I don't
I
know.
have one more slide that I'd like to discuss sometime.
Okay, yeah. I mean, you could share your screen and show it too. I mean, I know it's a little hard to navigate the.
Well, yeah, I can do that.
Okay.
You,
Yeah, why don't
want
we?
me to do that now?
Yeah, why don't we start?
Okay.
And you can do that, and then I'll go into the rest of it.
Okay, well, I added a few things. This is something I liked from Wikipedia, and it shows the simplest 3D form of tensegrity,
Yeah.
and this is very stable. I actually was able to put it in a finite element analysis program, and it works.
All right.
Anyways, so there's strings and strings which are tension element and did you call them bars or rods? Rods or bars are the compression element, and they're in green here.
Yeah.
And then you had a 2D kite form, which is nice for studying the math and just as a beginner study, because the 3D one is difficult to put in a program, but also impossible to do by hand. So
Yeah.
yeah. And then I added you had this about whether it's static, determinate or indeterminate,
Mm-hmm.
and it has to do with the degrees of freedom. So if you attach a bar to strings, it only has itself that is moving, but if it's got if it's like this at the side here where there's 2 degrees of freedom, if you have 2 bars connecting to each other, then you've got well, if it's unconstrained, it's a double pendulum, and that's a chaotic system. But it is constrained by the strings, so I don't know, I've got I had this. This is a double pendulum, and it depends on the initial state as to where it moves, but it moves all over the place.
Okay.
What else? Okay, there's kinematic. It's defined as the number of independent zero energy deformation modes of a rigid body. And then there's the formula. And this is so you can do math with it.
Right.
Yes. And then this is the self-stress. So the members naturally have a stress on them. And definition of self-stress. It's the number of independent states of self-stress mechanisms. I don't know. A structural system with S equals 0, it means the structural system is statically determinate. And I had the reference over here. GAN 2020 Computational Modeling of Tensegrity Structures. So both main parameters of K and S can be calculated after determining the rank. So you can actually get a matrix for this. And so there's these 4 classes of tensegrities, and they've determined whether they're statically determinate or indeterminate, and kinematically determinate or indeterminate. And you don't go very far before you can't figure them out. If they're indeterminate, the math doesn't work for you anymore. Like the matrices go off to infinity. So that's why they often say that a tensegrity only has strings attached to rods, or rods attached to strings, and you don't have 2 rods attached to each other. And then that gets back to the double pendulum idea.
Right.
So, and then, ta-da, there's another K. So this is another K. And if you take the elasticity and area of the string divided by the area and elasticity of the bar over each other, you get another K value. And if you play with that, you can change the slope of the stress strain curve. So that's how I match the stress strain curve of real things like skin or a thin layer of cells to a tensegrity for my thesis. Like I could actually change the order of magnitude of the graph by changing these.
Really?
Yeah.
This
That was a major discovery. Then I could do my thesis because I could match it to other people's data.
right.
Anyways, you
What
can
is-
see the elasticity of the bars is 200 GPa, which is like steel. And this is according to Dr. Ingber and other people. And the strings are also fairly they have a high elasticity, so they're fairly stiff. And they've got 100 GPa, which is like steel again.
Yeah,
Not
yeah.
quite as high as the bars, but close. And even 0.5 megapascals is a lot. So in a cell, the strings are fairly stiff. Anyway, that's what I wanted to show you. If you want to see my thesis presentation, I can show you how I matched things to skin and the layer of cells. You want to see that?
Yeah, that'd be good.
Yeah. Anyway, so this is my thesis, and I started off with my introduction. So this is epithelial tissue, and it has a certain structure. It's different from the interior tissues, which fit together more like bubbles, and they're more of a hexagon, not a hexagon, hexad, I don't know, 3D shape.
Yeah.
Anyway, these are more flat, and they're attached by their corners and the bottom of the structure. And this is a stress strain curve, and this is for skin. And it starts off kind of disordered, and then you pull on it and becomes ordered, and then it breaks here. So this is the stress and strain curve for this.
So it undergoes a phase transition, then a failure. or critical failure, I guess.
Yeah, it has two slopes basically. Has the bottom slope here, and then this steeper slope. Yeah, it's phase transition, and it's got a J curve.
Okay. Yeah.
This doesn't always happen with tissue. Sometimes the curve is flat. Anyways, this is showing the belt that's around the top of epithelial cells, and they're not square. Occasionally they become square. They're very mobile, and they're more or less a hexagon shape, as maybe you can see in part C here.
Yeah.
But they all have this belt that's around the top, and it's a tight junction and adherence junction, and they are solidly attached. This shows the actin and myosin fibers that are part of the belt, and they're actually mobile. They can tighten. And I'm not doing this slideshow properly, but oh well. And then there's trijunctions. So the cells at the corners are held together with trijunctions, and they go into the cell somewhat. They've got a thickness to them, and this is important for the behavior of this cell sheet. And then this is my model. So I made a model with a top ring and with attachments to the bottom, which I called the basement membrane. And then I had the trijunctions on the corners where it attached. And I don't know if you can see this from here, but I had posts in here to hold my model up because I didn't get more than 3 cells done. I had a lot of trouble with this. I couldn't get it to be stable for a finite element analysis program. And the way I did that was I made this trijunction in the middle different from the ones that were attached to the posts. So this is the model build, and this is a 3D tensegrity showing this is in tension and compression. And I put these at the posts to hold the structure up, or pretend to be the rest of the cell sheet, since I only had 3 cells. And this more complex trijunction was used in between the cells in the middle. And that's the only way I could get the thing to stand up in a stable manner for the finite element analysis program. So it was very frustrating to find that I needed this structure in the middle and the other structure for the sides. I don't know how I even discovered it.
Yeah.
Yeah. I think I had professors going, make it work. But anyways, so this is a hexagon that I built for the 3 layers. And then my building blocks were this type of a tensegrity for the sides. So then you get a hexagon with the tensegrity sides. And when I made them opposite to each other, you can see I've got one going this way and another going that way. Then my model worked. If you made them both go the same way, then the model didn't work, but it showed some interesting properties.
Right.
Yeah. So how many countless hours I spent messing around with that. And so eventually I got my model. Like I said, there's this T4 trijunction in the middle, and the T3s were on the sides, and if it did anything else, it did not work. And yeah, so these trijunctions are small compared to the rest of the structure, but very important. So I've already got this at a micrometer scale, and then I put these trijunctions in, and the program was more or less going, "What are you doing? This is too small." I'm not supposed to think of a program having a personality, but in the end, I did. And these are the 3 models I used. These are cuboidal, and these are squamish, and then you had columnar ones. So the properties of each of these, even though they're basically the same structure, just different heights, had different properties, which also was interesting, but our epithelial cells come in cuboidal, squamish, and columnar, so. And I could have played with the heights of these some more, because the properties did change when you change the height. I guess that it's kind of obvious when you think of it, but
Yeah.
anyway. Yeah. So boundary conditions, I attached spring foundations to the bottom, and in the next, and then I had to have some pinned points. So on these posts, I put pinned points. That's just so the model would work.
Right.
Yeah, and so I could change the spring foundation. I could change the pre-strains for the bars and strings, and I kept them the same. Like that makes mechanical sense to keep the bars and strings at the same pre-strain. And then I did a range of forces on the middle of on the middle part of the structure. Did I show that? Yes. Okay. I applied the force to the top and the middle at the 3 top points of the middle of the structure. If you zoom in on it, there are 3 points here.
Okay.
Anyway, that's what I did. And then I used a range of spring foundations, range of pre-strains, a range of forces, and then I had to do the whole thing by hand. I had to individually apply each one of these forces to the middle. I couldn't automate it. Like usually you automate it by telling the program, "Oh, go between this force and that force and draw me a graph." Well, no, I had to do this individually and then draw my own graph. I might have been able to hook it up to MATLAB. So the MATLAB would have drawn the graph for me, and I just never mind. I'm just going to do this and be done with it, instead of messing around with the program, and I would have had all of my data by the time I'd figured out how to use MATLAB with console.
Right.
Console. Yeah. Okay, so this is, I was measuring engineering stress, and it's on this top triangle on these 3 points. and I should have had that. I should have had this earlier. I suppose it would have flowed better, but I didn't. And then I did engineering strain. So I measured the change in height to the initial height. These are the results here. So overall observations, the 3 cell tissue model was influenced by the values of the strings and bars and the connections. So that was the cell-to-cell connections at the trijunctions and the cell-to-basement membrane attachments. And this occurs in other models of cells. And so I had sample graphs, and it's backwards to what you think, because we've got, this is tension, and it goes from here downwards. This is engineering, so this is flipped on its head, because tension goes down and compression goes up. Anyways, what I was looking for was a separation of these curves for different pre-strains. In other words, the cell has control over its environment if you have, if it can pre-strain itself. And it does, because it's got the actomyosin belt, and it can tension that. So it can have a different pre-strain, and so it can act differently to force. So there can be a different displacement curve for different forces. And then this is, I've already showed you the values for K, and then I had a couple more. These are the cell-to-cell connections. These are 2 compression curves, and this is compared, I should have labeled this better. This is a squamish cell, and this is a squamish cell. This is the results of changing membrane width from 0.2 to 0.5 micrometers. Okay, so this is 0.12 micrometers in depth, and this is 0.5 micrometers in depth. So you have to have a certain width of the top part of the cell, like the membrane has to be wide enough for a separation of these curves. This is basically a fail. The only thing that's different is something without pre-strain, and the rest of the pre-strains kind of end up on the same line. So a cell does better in this environment. So the membrane of 0.12 micrometers works better than 0.05 micrometers. I thought this was an interesting result. This is a very good separation of curves here. And this is a columnar model. So columnar models work very well at certain pre-strains. And the cell to basement membrane attachments also have an effect on this. Let's see. Sorry, I didn't go through this before I presented it.
That's fine.
It's been a year.
Yeah.
Okay, the pre-strained curves become more linear, and the pre-strained zero is separated from the other pre-strained curves by a lot, increasing the elasticity of the zero pre-strained curve. Yeah, okay. This could represent a diseased state. Like this curve here could represent a diseased state.
Yeah.
Anyway, this is what I wanted to show you. I actually found some data for single cell sheets, which is more or less what I was modeling. And I did manage to match my tensegrity model curves to the curves for this. These are more J-shaped curves than what I ended up with, which was more or less a straight line. Is a straight line, but the curve that matched these best was again these oriented cell sheets, single cell sheets, and these were the tension and strain were measured at 90 degrees to how the cells were laid out. So these cells are laid out. These are not oriented, but then you had oriented cell sheets here. And you can, I don't know if you can see that, but they're lined up like this. So these curves were measuring the stretch going this way. And then the next ones here were measuring the cells going perpendicular, like they were stretching them perpendicular to their layout, and I got very good results for matching my model to this strain curve. And the main reason why this was a good model was because it covered all of the strain. It went from 0 to 40, or it's 0 to 50%, and it just matched these curves. Like the tissue broke at these points, and my model went to that and beyond. And usually the models only get up to 10% and then they deviate. Like they don't match curves. They don't last that long. For instance, something like an Ogden model, if you've ever heard of that. And this was an interesting curve, or this is the actual data, and it was all over the place. But it kind of followed a pattern, and this is the pattern. And I was able to match it with my model. And it was interesting because the 10% pre-strain is here on top, and the 45% pre-strain was on the bottom. And this is all in compression. And it's opposite to how this normally works. Normally you have 10% on the bottom and the 45% pre-strain on the top. And my model actually did that in some instances. So that was interesting as well. I could get it to flip from 10% on the top and 45% on the bottom to having 45% on the top to 10% on the bottom, just depending on the parameters I used. So I could play with it that way, which was a surprise. And then I compared the human skin tension, used a cuboidal model and a squamish model. So the cuboidal model ended up here, and it's in the dotted line, and the squamish one is the green one here. It's kind of in the middle. And that's when I changed the K value I was talking about, so that the curves were kind of matching what was going on. They still don't have this J curve thing going on. My model only did straight lines, and that was too bad, but considering it was made out of sticks and rods and there were only 3 cells, it was it was rather simple, we'll say, compared to a bunch of thousands of cells, say in skin. That's why this model of single cells was a more accurate thing to compare it to. Anyways, I had conclusions, but if you want to see them, so I can do my model work to match stress strain curves of actual real tissue. Main conclusion is the tissues, that like tissues, the model is reliant on their attachments and the values in its structure. In other words, the elasticity value of the strings and rods. And my model modeled beyond what other models were doing, like the Ogden models. The Ogden model can do up to 20%, and most tissues yield at about 50% strain, so they're not covering the full range of the tissue. And my model covered that. So they said it was a done deal, and then I'd proven that I'd actually modeled something.
Okay.
So, any questions?
Yeah, thank you, Susan. That was good. I had a couple of questions related to some of the things that you talked about. One is that you mentioned pre-stresses and pre-strains, and then what does that mean in terms of the biology? What would be an example of a pre-stress, and what would be an example of a pre-strain in biology?
Well, muscles naturally have tension while you're alive, like they're normally tensioned. So that's like a pre-strain, like it isn't totally flaccid.
Okay.
And there's squamish epithelial cells covering most tissues, and so they're holding an organ, and those are naturally tensioned. If they aren't, that means it's broken. So that's a couple of examples. Tissue also naturally takes that trijunction that's in, or the belt of actomyosin that surrounds them. When they're a epithelial tissue, they'll have this belt at the top of the cells or the bottom of the cell, actually, and they can contract that, and that'll cause a tension, and also you get waves of tension and pulling throughout cells. It just occurs when there's movement.
All right.
Does that help?
Yeah. So there's a dynamics there. It's not just like a single measurement. Like, we tend to think of these things as like, I'll just take a measurement and this is the sort of the output. I mean, it's nonlinear, but it's like, you'll just think, well, that's what it looks like if you just kind of test this one little structure. But we're thinking about tissue where we have all of these different things interacting, maybe these different tensegrity structures interacting, or larger tensegrity structures connected to other things, and that produces some interesting dynamics.
Connect, like if you have ligament attached to bone, for instance,
Right.
and that's attached to muscle, so you're always have some sort of tension going on.
Right. So you get waves,
Okay.
and then the waves kind of go through the tissue.
There's
And,
waves.
yeah.
Yeah, I sent you a paper about waves in the beetle this week. It says spatial-temporal segmentation of contraction waves in the extra-embryonic membranes of the red flower beetle.
Okay.
I sent that to you, and I was hoping that Dick would see that. Because they said, we don't know what these waves do.
Yeah.
Causing cell differentiation.
Right. I didn't pull that up for this week, but yeah, it's we'll have to go over that.
Okay.
Yeah.
Okay, well, I'll stop sharing.
Okay. Yeah, so that's interesting. And then, we have these different sort of, use, or I call them use cases, but real world implications of these models. So, we have tensegrity, which is, of course, we could describe a lot of things as tensegrities. They exist within a tissue, and then they have these dynamics. And yeah, it's like, we have like studies where they measure the stresses and strains in some system, and then that's the data that we have, right? Like, we're not going to get better data. We're not going to get like real-time data where it's like just measuring things as
the as far as I know, if you haven't measured it in stress and strain, it doesn't exist for an engineer.
Yeah.
It doesn't mean anything.
Yeah, I know. So okay, so yeah, I think that's good for now. Yeah, thanks for adding to that slideshow again. Like I said, we'll be going through the slideshow and adding in things and like kind of crafting it down to something, and then we'll go from there. I just wanted to get a lot of that in one place and kind of think about what the core sort of mathematics are, what the core ideas are that we can distill down. And
I still don't understand the math.
Yeah, well, it's
for this because they do this in several different ways.
Right, they do have different methodologies.
And I did have, and I messed it up. I did have a paper about tensegrities with non-stretchy strings, because according to Stephen Levin, our cells or the biological tissue don't have that much stretch in them, and a proper tensegrity is more like a string and it's not an elastic.
Right.
It's a string. And so they don't stretch very much.
Right.
So I had that article about working with strings that didn't change in length and that were not that elastic. It's around here somewhere.
Right. Yeah, so that brings up another question, which is that tissues are anisotropic, which means that they have different, like the different parts of the tissue have different properties. So you have, like, say, for example, muscle and bone, and they have their own sort of physical properties. And so in your modeling, in a tensegrity, you're kind of breaking that apart and saying this tissue type is, or this cell type has a certain sort of set of properties, and they fit into the tensegrity structure. But it's I don't know, is that, like, do you think that's a problem, or do you think that's just kind of baked into the tensegrity model?
You have to model them from the actual structure of the tissue. Like that, why I chose epithelial tissue. It's fairly simple, and it's a single. It's not a single layer, but you can do a single layer. There are some epithelial tissues that are single layer. If you look at the structure of epithelial tissue, say of skin, it starts off with with things that sort of look like my model, to a deeper structure, and they're more of the dodecahedron. That's what they are. Right. Dodecahedron type structure. So it changes as you go in depth, and then you have to look at where they're attached to each other at the trijunctions, or whatever they're using for attachments to each other, and then you have to model that.
Yeah.
Yeah. And the tensegrity structure inside does change, I believe. It's just where it is at that moment in time.
Yeah.
And the simplest tensegrity structure I could to get a hexagonal 3D structure. So it might be more complex than that. I don't know. I tried to change that and add more structure to it, and it just fails. It falls over.
Right.
Yeah. And I put extra, physically extra bars and strings in the middle, trying to mimic the top and bottom of the cell, and it didn't add, like it just did not add to the attachments. But that's probably a fault of the program, but maybe it's also a structural thing, and I never got to check it out. Like, they wanted this thing done by Christmas.
Yeah.
Okay, then here it is.
You can only do so much, yeah.
Yeah, but I put extra stuff basically on the top, on the membrane of the cell, and the only thing that made a difference was the depth. And I could put all kinds of different things in the middle, and it didn't make a difference to the properties. Like, it maybe started its linear path sooner or later, but it was basically the same curve.
Yeah.
Yeah.
Okay. Yeah, thanks. Thanks for that. All right.
You can get these things cheaper, but
yeah.
Sort of.
So it looks like we have Barshan and Shreyas. Welcome. I don't know if you wanted to introduce yourselves or.
Yeah, hi, am I audible?
Yes.
Yeah, hi. So yeah, I did have a chat with Jayadrat, and I worked on things he suggested. So yeah, we can discuss that if you want.
That's good, yeah. Sounds good. Um, Barshan, did you want to say anything? All right, why don't we move on to some of the things I wanted to talk about. So this is a commit to DevoGraph. This was something that was committed this weekend. So we had add-based graph with HyperEdge support and tests. This was by Ashu Rajput and or Ashu Rajput 08. And so this adds some functionality to some of the things that are going on in the DevoLearn, in the graph neural networks repository. So again, this is just adding to the capabilities of that software. So thank you for that commit.
Hello, sir. Am I audible now?
Yes.
Hello.
Yes.
Okay, so I'll give myself a introduction because this is my first meeting. So I'm an undergraduate in my final year, and I'm currently pursuing my bachelor's in technology. So I went through the past two weekly meetings, and I also went through the previous year's recap. So what I got from that is a very good sentence that they said in the last, that a living thing is much more than a sum of its parts. So that like sums up the aim for the next year. So I guess we are looking more towards the other outer forces which influence the living being than just the embryo and its inner organs. So taking this into account, I also went through a paper which uh, looks into Markovian dynamics, and I feel like that would be a great uh, paper to work on. And keeping that as a foundation paper, we could go forward with that. So I also prepared a kind of an overview as a presentation. If Bradley sir, I could present that.
Yeah, if you want to do that now, you can.
Yeah. Okay. So it's actually very interesting that I went through it because I'm very new to neuroscience, and uh, taking control of an organism is kind of a big deal for me. So I'll just present my screen right now. All right. So is it visible?
Yes, it is.
Okay, so this is the paper. So Markovian dynamics for C. elegans and its behavior across scales. So these are some of the people who worked on this, and I'll just go straight to the paper. So the problem that they went through was that living beings move in complex ways, and it's hard to connect the tiny body movements, like which takes in milliseconds, into the long-term movements like finding food, which takes like minutes for them. So they've created a model called the Markov worm, which predicts the worm's future based on the previous movements or the current movements or the sequence of movements it has been through. So this is kind of a picture. I'll explain the top right corner of it. So what this is it breaks the movement of the worm into something called eigenworms, which is like numbers which is used to represent the movement of the worm, and they want to break it into five uh, coefficients, we can say. Uh, so this is one part of it, and let's talk about the data which they used. So they recorded like 12 worms for 35 minutes each, and they captured around 33,000 frames. So, so a snapshot of the worm is not really any use. So we need to see the whole movie clip to know where it's going to be in the next second. So they took like a sequence of 11 frames, which they used to feed into the model to predict the next frame. And these sequences were called microstates. So as you can see, this small picture. So here AT plus K refers to the uh, building blocks of the sequence, and they're like the single body shape at one moment of the worm. And the whole xk of t is the collection of the shapes over 11 frames, which is then inputted into the worm model to generate the next sequence. And after that, we have this, which is like the calculation of the probability of the worm while it's switching from one state to the another. And let's say, if for an example, if the worm is curling left state, what are the odds of it switching to straightening out or curling harder than it already is? So, using this model, we can of course generate a virtual worm which behaves like the real one. And when we are plotting this movements, I'll show it. So it's like a U map. We plotted it out, and this small dot is like the movement of the worm, and the blue refers to the forward runs of the worm, and this bluish and orange splints, they refer to the uh, turns or reversal of the worms, like if they're turning. Uh, Next, we can go through the. So the main aim, like the main discovery of this paper was basically they invented a lot of new behaviors of the worm. Previously, it was just two famous behaviors, which was like running straight or like turning around from its place. So this new discovery revealed like seven new sub behaviors, which was like fast wide runs, slow dorsal runs, and five more, which are, I guess, told over here. Uh, Deep ventral periods and a lot more, which you can see. And this is the result. Uh, One second, yeah. So this is the result. So there was a problem in the uh, model, like when they were like trying to get this posture to the path, so how it's moving, like relating it to them. So because it was mostly specific on how the worm is bending and not where it is bending, or the you can say the friction related to it. So they also use the concept of resistive force theory, which Sir explained, I guess, last week. And they use these physics equations to also take into account the drag forces of the worm's body against the sticky gel, basically, which it crawls on. And they were successful in this actually. So this is the results where the blue line is the simulation of the worm, and the black line is the actual data of the worm, and they were pretty close to it. And these are some physics equations which they integrated into the model further. And so lastly, we have those behaviors and how they have made a behavioral tree, like how this runs, like more like a tree which, if this is yes and this is no, it leads to this kind of behavior. And so the paper is mostly based on the probability of like finding food based on the movements of this worm, and they were pretty accurate about the results. So, that's all mostly that I researched about for this weekend. And I think that this would be a very good foundation paper to move forward with the aim of this year.
That's very good.
That's how
Yeah,
we should.
thank you. Yeah, thank you. Very
Yeah.
good. Let's see. Susan had a thing in the chat here, which was this
It's
citation.
just a form finding method for strings that are basically non-elastic,
Okay.
inextensible. That's
Yeah.
what they're calling it. That's all.
Okay. Yeah, thank you for that. So yeah, back to Barshan's presentation. Thank you for doing that. Yeah, the Eigenworms paper, that was actually something that some collaborators of OpenWorm did a while back. And OpenWorm, of course, had its movement database, where they characterized a bunch of worm movements through microscopy and had a database, which I'm not sure what the status of the database is right now. But those data are out there. And the idea was to take the observations of movement in the adult worms, do the eigenworm decompositions, and then characterize movements from that. And that's interesting. Just kind of getting the movement, like decomposing the different movement data, because usually what we have, and this holds true for like movements in the embryo as well as the behavioral movements, is we just have like cell tracking or feature tracking, which is where you have like an image, and then that image is segmented, and you have like the components of the thing that you're observing. It could be the individual cells, it could be the body, and then you basically decompose those movements over time. So you look at the dynamics of the different positions, and you put that together, and you find like the things that describe those trajectories. Now, that's something that I think would be interesting applied to like embryos, because we have data where we look at embryos over time, we look at cell divisions, we look at like migration of cells, and it's interesting because I don't know if people have done that where they've taken different cells in development, tracked them and then looked at the movements and described them in that way, like where you have like a eigen migration or eigen. Yeah, well, yeah, but in this case it would be like eigen embryo or eigen cell. I don't know. But the point being is you can use that same methodology in development. It'd be interesting to know. It would uncover some new information. So that's good stuff. Yeah. So yeah, that's great. And then of course that that could tie into tensegrities, looking at sort of the stability of structures. I don't know. Well, I think they're.
We can integrate all of those into one.
Yeah.
Those forces.
Right. The functional modes in tensegrities. I think that's, you just look at the things that are stable, or if you do a simulation, you look at the things that are stable and unstable, and you're actually extracting eigenvalues in the matrices in tensegrities. So like you have these matrices of the different components of the tensegrity network. It's represented as some sort of matrix, and then you're taking components of the matrix, diagonals, rows, columns, and then getting the eigenvalues for that, and then it's, what does that describe? It describes different modes, soft modes, other modes.
Definitely computing tensegrity structures.
Yeah.
Yeah.
Okay. So that's good. What do we call it? A meeting then? Thanks for attending. Thank you for Susan and Barshan for presenting. Those are good presentations.
Yes, hi. Hello.
Yeah. Hi.
Yeah, hi. If we have time, I would like to also present. Jayadar told me, I had a chat with him as to what are the aims for this year and what he worked on, and I cleared my doubts with him. So he asked me to work on the Connectome data set. So I did work on it, and if we have time, should I present or should we push it to next week?
Oh, actually, we could do that. Why don't you go ahead and present? Yeah, why don't you do that?
Yeah, sure. So, wait just a second before we proceed. Yeah. So Jayadrat told me that I can find the intermediate state of connectome connections between two snapshots. So the data set is such that we are given snapshots of various connections like L1, L2, L3, and so on, which from the baby stage to the adult stage, and we need the the states between, we don't know the states, it's just two snapshots. So he asked me, he couldn't explore in that direction, so he asked me if I can do it. So I did go ahead and I got two papers. So one is a latent ODE for irregular sample time series. So what I observed is that just a second, I'll go up, and yeah, so we have 8 files. So between those files, the time series which we have is not equally sampled. So say there is the gap of 1 hour between L1 and L2, and then there's a gap of 8 hour between L2 and L3. L1, L2, I'm referring to the states as to so L1 and L2 are the states of the connectome. So for that, this paper I got, which is the latent ODE, so it's a differential equation solver, which can solve any differential equation. So why is it so? I'll just get to this in some time. And this other paper is a graph neural ordinary differential equations. So what this does is, so I'll go ahead with this paper first. Just in short, I'll give a one-line short intro of this. So what this does is, since connectome is a graph and it's not it depends on the neighboring connections as well. So this models the whole connectome as a graph, and in a temporal way, it can add nodes and so on. And then since each node has an internal representation with the graph neural network, so I combined both of these papers and tried to devise a method where the internal vector which is made, so that is evolving, and we find a differential equation to model it, how that vector changes from, say, state 1, L1 to L2 to L3 till adult. So if we get the differential equation, then we can just give any time step between L1 and L2, say some time step one and a half hour. So what is the state of connectome at the time 1.5 hour? So that's why I went in this direction. So when I went in this direction, yeah. So basically, what I'm doing here is, should I go through the code or should I just give a overview and then if any doubts, so I'll go in the code. So here I'm just preparing the data set, like all the tensors and all, to do the data preparation. Then I'm defining the Connectome ODE solver. So this is the ordinary differential equation solver, which solves the internal vectors for this. Like it models the differential equation for the internal vectors of the neural network. Now, one point, like the most important point is that, like this is the uh, just a second. Yeah. So this is a GCN conv. So this is what makes the internal vectors. So we have a graph convolution network which makes the internal vectors, and then the ODE function works on those internal vectors, and it uh, learns the ordinary differential equation for the internal vectors. This is a point to be noted. Like it's not like it learns for the connectome, it learns for the internal vector, which is modeled by the graph convolution network. Right. So we train this thing over like some 2000 epochs, and then this loss converges. I was a bit surprised as to why the loss converges so steeply initially and then quite slow later. But yeah, then here we do the missing frames prediction, like the interpolation. I just sample like a bunch of, like, say, time at 0.5, 1, 1.5, 2, 3, and so on. So how the thing is a model is that I'll just go here. So we have say we have approximately 220 total number of neurons. So from like zero, it starts with like some less number of neurons and it goes on increasing. So what I did is, like, I created a matrix where all the neurons are present, like 220x 220, and then all values were zero initially, right? And then whichever neurons were active, like since the number of neurons were increasing with time. So initially all were zero. Those who were getting active were turned on to one, and then accordingly things were trained. Right. So this is a general overview of what I did. I didn't go into the math and the loss function and the like training part of it. For a matter of fact, I used binary cross entropy with logic loss. So like, yeah, binary cross entropy with logic loss for this model. And yeah, so this is the connections. So this is like connection between, okay, one more thing, I'll just come. So this is like here what we're doing is, say there are connections between various neurons, right? Say I have n number of neurons, so n cross n I can have, in theoretically, in mathematically, I can have n cross n ncn connections, right? Each neuron connected to other neuron. So this, which actually doesn't happen in biology. So this is like the connection probability as to what neuron can be connected to some other neuron. So that is the graph that is the heat map which I'm plotting. So this is the heat map for it, and this seems continuous as to, like, it seems like, okay, fine. However, the, like, initially the blue connections here is quite less, and here it increases, and so we can see that the connections are increasing in a natural way. But I don't claim that these results are quite accurate, because this is a preliminary like development by me. Like I just took two papers, I found papers, and I tried to merge it and get a new method. But yeah, this is a preliminary examination for if the method works. I'll get input from you and work on this further. So yeah, this is what I did. And one more thing, just to end, like the last note, that since the connections are sparse, like the matrix which I saw was quite sparse. Like not every, since NCN is a very large number, but the actual connections are quite less, right? So for that, what I did is, in the loss function, if, say, the model predicts some connection which is there, like say the connection is 1, but it predicts it to be 0, which is off. So then the loss is quite high. But if some connection is off and it predicts on, the loss is like sort of normal. But if the, like, if it predicts wrongly for the imbalanced class, then the loss is quite high. So that is the way through which I tried to balance it. So like, this is the positive weight for loss, like 5 was the positive weight. So it's like 5x loss it will incur if it predicts a imbalanced class incorrectly. So yeah, any questions?
Yeah, thank you. Thank you. That was great. Yeah, I like this approach of using ODEs to describe the connectome evolution. So I know that last year we did temporal graphs, just the temporal graph outline, and then, you know, having the different stages. And now we have like ODEs that kind of fit the dynamics together. And I know, like, in Saturday's meeting, I know Morgan had talked about people doing like stuff with neural differential equations and how that's sort of a newer field. But yeah, this is good. This is good. Thank you. So that's again using Connectome data, so we can look at the dynamics there. All right.
Right. So, what would you like from your end? What can be the improvements? Like, what do you think should be done in a better way or modeling? Or any input from your side so that I can work for the next week on that?
I don't know. Just work on improving the technique, or maybe doing a couple of demos where show kind of the connection between the data set and the graphs that you're producing. So you produce like the heat maps and things like that, but it's hard to see kind of where that's coming from and understanding
Right.
what the code is doing. So
Okay.
then of
Yeah.
course go through and optimize the code and kind of figure out
Sure.
if what exactly is the key thing that we should take away from it. How does it perform? I don't think you can really. I mean, I guess you could vary things in the data set, but just to show kind of what the power of this method is.
Right.
Yeah.
I get it. Understood. Okay, I'll work on this. And one more thing I was thinking, which I'll work on, is that like there are no labels, right? I'm predicting what is the connections between two time frames. So there are not many labels to us to see if it is correct. So is there any way through which we can see if the outputs which I'm getting are actually that good? Now what I'm seeing is if the interpolation looks systematic, like the transition looks smooth, is what I'm seeing. But
Right.
I want some like grounded proof that okay, the thing which I'm doing actually makes sense. So
The
what do you think in this context?
well, the data set that you're working from, doesn't that have labels, like in terms of if you're looking at different synapses? And some of the data are just kind of counts, so those aren't technically labeled, but some of them also have like specific cells that the synapses are associated with, or pairs of cells. I don't know exactly what data set you're plotting it out with. That's kind of one of the things that we kind of need to figure out. Like, go back and look at the source data and say this is the input, and then this is the potential label, or this is the potential thing that we're putting into the model. And then we can kind of, because we can create labels sometimes if we know kind of where the data is coming from. Like, we have other data sets that we can use to sort of augment what we have, so we create labels that way. So yeah, there are a lot of things we can
Sure.
do, but I need to know kind of where things are coming from, or we need to know that.
Sure.
Well, thanks for doing an overview of that. That sounds interesting. I'm kind of interested in your orthogonal lab talk, like I saw it.
Oh,
And it was the paper you presented a couple of weeks ago, next generational stem cell models for studying brain development and disease.
oh, okay. Yeah. Oh, maybe that was this
Yeah.
last week. Yeah,
Was okay.
maybe.
Anyway, no, I just I tried to look some of it up online and couldn't find it from the Sloan Kettering Institute for Cancer Research. Anyway, some of what Shreyas just talked about kind of fits with some
Okay.
of the
Yeah.
neural
Just
models.
send me the title and I can get it for you.
Okay, thanks. I have trouble getting things sometimes, so
Right.
maybe I'll just send you a couple of my problems.
Yeah.
I put them in Word. I put the title in Word and couldn't get the paper. So
Yeah.
all right, and
All right.
the flower beetle things in the chat there.
All right. Yeah, that paper.
You took note of it, so
Yeah. All right. All right. Well, thanks for attending, and thanks to Barshan and Shreyas and Susan for presenting. All right.
Yeah.
Have a good week.